Today we'll be using the gene bioextraction kit to extract DNA from our samples. So the first thing we're going to do is we're going to prepare our samples. Make sure it's mixed. If it's a liquid or if it's a tissue sample, you make sure you add it into your solution. First thing we're going to do is we're going to add 400 microliters of our sample into our test tubes here. It will differ between samples, so consult how much you should add per sample. So, and now our second sample. So the first step is you're going to add solution DS to your Eppendorf tubes. So you're going to want to add 200 microliters. So you're going to want to set your pipetter to 200 microliters. You want to add them stepwise like we've been doing. It is important that they are homogenized, so we're going to use a vortex to homogenize them together. So you're going to want to vortex these, make sure that they're properly thick, so shake them up and down, vortex again. And now the other sample. Now, since we've covered that step, there's an optional step you can do. If you want your solution to be free of RNA, you can add RNase at this point. So I'm going to grab some RNase. And this, as you can see, has been on ice, so it's cold. And then we're going to add 4 microliters to each sample. So we're going to grab our pipette tips. We're going to add 4 microliters to each sample. Because it is a small amount, you want to be sure that you are in fact adding it to your solution. And make sure to switch pipette tips between. And now the second sample. Okay, so now you're going to want to incubate these at 5 minutes at room temperature. So we're going to return our RNase to the side and we'll start our timer for 5 minutes. Alright, so now that the timer is finished and we've waited the incubation period for the RNase, now we're going to add 20 microliters of proteinase K and 220 microliters of solution MS. So we're going to grab solution of proteinase K. This was also on ice. It's a good idea to keep yours on ice if you need it. We're going to grab pipettes. Now I'm going to do it for the second one. So I'm going to open this one up. And then you're going to add 220 microliters of solution MS. So we're going to first return protease K back on ice. We're going to grab our solution MS, and we're going to add 220 microliters to each. So we're going to grab your pipette tips. Now that we've added both solutions, we're going to vortex to make sure they're homogenized. Support with every step to make sure everything's mixed together well in order to make sure the solution is homogeneous. This will help make sure everything goes to completion. Now that we've vortexed every single one of them, we're going to incubate them at 65 degrees Celsius for 10 minutes. So we're going to add them to our hot plate here. And we're going to start a timer for 10 minutes. So now that the timer has expired, we're going to remove our samples from the heat. And in the meantime, we're going to swap this out for a mini centrifuge. Alright, so now that we've done that, now we swapped it out, before we put it into the microcentrifuge, we're going to add 220 microliters of ethanol into each. So we're going to do that before we start our whole centrifugation process. As always, we're going to vortex between steps to make sure everything is homogenized. Put the ethanol away. And then we're going to transfer these into these centrifuge tubes. They're 2 milliliters. And we're going to use them during centrifugation to collect any excess out that we don't need. So what we're going to do is we're going to take 800 of each, and we're going to put them into these tubes. So set your pipetter to 800. Now that we've added the ethanol, we're going to use these spin columns, these DNA extraction spin columns. The column inside is going to catch our DNA, and we're going to use it to extract it. And any of the flow through that we're going to discard is going to be inside this tube. So the first thing we're going to do is we're going to transfer our samples into these. So we're going to take 500 microliters of our sample, and we're going to transfer it to the capacity as much as we can. So we're going to vortex it, make sure everything is mixed, and make sure any precipitated DNA is going to be added to the columns. capacity of these columns are 500 microliters, so any extra will be done in the next step. We're going to repeat the same step over to make sure we extract everything is collected. So load this up to 500 microliters. Close this out for now. Then we're going to go to our second sample. Vortex to make sure everything is homogenized and any precipitated DNA will be collected. Next to the second spin column. Now we're going to spin these columns at 12,000 RPMs for 1 minute. So we're going to open our mini centrifuge, remove them, and we're going to ensure that we place our plates opposite to each other. So we're going to place one in the 1 position, and the other one is going to be placed at the 7 position. They should be opposite each other, so they offset each other. And now we're going to let this run for 1 minute. Now that we've spun them for 1 minute, we're going to collect our spin tubes here. The flow through is not what we want, so we're going to discard the flow through. So you can simply do that by removing the column, making sure not to make contact with the flow through. And we're going to repeat this step again to collect the remainder of our sample. So again, we're going to load this up for 500 microliters. Why is it? Oh. Get a vortex before you collect the sample. If your sample is less than 500 microliters, just collect as much as you can. Now the second sample. This one, it's a little easier if we open it with 2 hands. And now we're going to repeat the same step once again, making sure they are in opposite sides to stabilize. And same thing, make sure you put the cap, for 1 more minute. Now that we've collected the DNA spin tubes, we're going to discard the flow through once again. We don't need them. At this point, our DNA is trapped inside the column. At this point, we're going to be working solely with our column here. So we're going to do a series of 3 different wash steps to purify our DNA, making sure that we wash out anything that we don't want. So the first thing we're going to do is we're going to add PS buffer, and in each wash step we're going to add 500 microliters of our buffer to the column and spin it for 1 minute. Same conditions, 12,000 rotations per minute. So we're going to open our centrifuge tubes. We're going to open our PS buffer. making sure again that we put them at opposite sides. So in this 12 position plate or spin, we're going to put in positions 1 and 7. Although it doesn't matter where you put them as long as they're opposite to each other. Same conditions as always. I'm going to start the spin. Now that we're done with this, we're going to collect our spin tubes. Again, discarding the flow through as that's not what we're after here. So we're just going to discard the flow through. And we're going to do to the second wash. This time we're going to be using a different buffer to wash. We're going to be using the PE buffer. So carefully discard. So we're going to be using our PE buffer, same as always, same spin conditions, same amount, 500 microliters. Bring these up. And now we're going to spin these once again. All right. One final wash step. As always, discarding our flow through, because at this stage it's not our DNA. Using the same buffer we used as the last step, using the PE buffer. So we're going to close this as we're done with it to the side, and we're going to run our columns one last time, last wash state. Now that they're done, we're going to remove them from columns. And at this stage, we want to be very careful not to splash, because we're trying to make sure all the flow through is at the bottom here. So we're going to discard the flow through. Discard the flow through. And now we're going to do one last step. We're going to spin it for 3 minutes this time to ensure that it's dry ethanol. So we're going to spin it one last time, this time for 3 minutes with the same spin conditions. And let's set this to 3 minutes. Alright, so now that we've done the 3 minute wash, we are good to go with the wash steps. Now we're going to be very careful not to let the membrane touch any remaining liquid. And at this point, we're going to discard the flow through and as like the tube. And we're going to transfer it to a fresh tube. So we've labeled this tube C for sample C. And we're going to remove the column slowly and put this into this new tube mark C, and we can discard the column at this stage. We're going to do the same thing with sample P. We want to go slowly, make sure we don't touch the sides or anywhere where ethanol may be. We can discard this and into a fresh tube. And now what we're going to do is we're going to elute our DNA. So these are our old sample tubes we don't need. And now we're going to use elution buffer. We're going to use 100 microliters of the elution buffer over here. And we're going to want to try to put these at the center of the membrane. The reason being is we're going to leave this to incubate in the center for 2 minutes, so we want all of it to be in the center over here. So we're going to open this one and grab our pipette. And you're going to be very careful to try to put this in exactly in the center. Swap out your reduce contamination. We're going to do it again for the second sample. So you're going to want to be very careful. So if it touches anything else, you want to discard it. Because at this stage, anything that does elute through will contaminate the DNA. And we let this incubate for 2 minutes. Now we're going to spin the columns one last time to ensure the inner column is closed. You don't want your DNA to contaminate it. So make sure the settings are the same settings we've been using the entire time. So make sure the inner column is closed. That's the most important one. The outer column probably won't close, but that's alright. Inner column, make sure they're staggered and the open column is at least going into the center. Now we're going to close it, make sure that they're opposite of each other. And we're going to spin it for one last time for 1 minute. So now that they're finished spinning, what we should find is at the bottom of our tubes, they now contain liquid. And if you've done this right, at the bottom should be your purified DNA. So now you're going to discard your column, and you're going to store this on ice at -20. You can do the same thing with the other sample. You can discard your column now. And this should be your purified DNA, which you can do with further analysis or further studies with.